Hello friends, welcome to the channel. In this video, we will discuss about the most widely practiced staining technique in bacteriology, the Gram staining. This staining technique differentiates the bacteria into two major groups, the Gram-positive bacteria and the Gram-negative bacteria on the basis of color retention by its cell wall. The Gram staining is a differential staining technique developed by Dr. H. Krishnan Gram in 1884. The technique identifies and classifies the bacteria into two major groups, Gram-positive and Gram-negative. The bacteria which retain the primary stain and appear dark blue or violet, that is not decolorized after the staining procedure, are called Gram-positive, whereas those that lose the crystal violet and counter-stained by safranin, that is appear red, are referred to as Gram-negative. Next comes the requirements. So the requirements of the experiment are 24 hours or less old bacterial cultures, crystal violet stain, grams iodine solution, 95% ethyl alcohol, safranin stain, distilled water, droppers, Bunsen burner, glass slides and microscope. Now let us look into the procedure. So the first step is preparation of smear. For the preparation of smear, place a drop of distilled water on the slide. Now add the microbes to the liquid and spread it over the slide. Now air dry or heat gently. When dry, briefly heat fix the cells to the slide. For heat fixing a slide with the specimen, pass it over a heat source such as a flame several times using forceps. The slide should be passed very quickly through the flame and not be heated excessively. In the second step, hold the smears using a slide rack or slide holder. In the third step, cover each smear with crystal violet for 30 seconds. In the fourth step, wash each slide with distilled water for a few seconds using wash bottle. In the fifth step, cover each smear with grams iodine solution for 60 seconds. In the sixth step, wash off the iodine solution with 95% ethyl alcohol. Add ethyl alcohol drop by drop until no more color flows from the smear, but not exceeding a time interval of 10 seconds. In the seventh step, again wash the slides with distilled water. In the eighth step, counter stain the smear by applying safranin to smears for 30 seconds. In the ninth step, wash the slides with distilled water and blot dry with adsorbent paper. In the 10th step, let the stained slides air dry. The 11th step is observe the slides under the microscope. The observations of the experiment are: the bacteria which retain the crystal violet stain appear purple in color, while those that got decolorized with ethyl alcohol and retain safranin appear pink in color. Now the results that we get in the experiment can be explained as: the bacteria that appear purple are referred to as Gram-positive, while those that appear pink are described as Gram-negative. This is how the Gram-positive and Gram-negative bacterial cells are seen under the microscope after Gram staining. So this is all for the day today. Thank you.